That was quick. You guys must be ready to dive into biochemistry today, I'm guessing. Yes! It's Sarcastic Friday, isn't it? It's opposite day. Okay, so note cards up here for the final. I will also have them in my office. Please, one note card. And as you know, you can fill out both sides. You can fill out the edge if you can see what you're writing on the edge. You cannot put anything on the card. So you cannot tape something to the card, and you cannot print something on the card. It has to be all your own handwriting. So everything on there must be original from you and your hand. And you have to bring a card to the final. If you don't bring a card to the final, then you will get docked, and you get the card from me. So those are important points. There's a reason I do it, not to just be a pain in the ass, but there's a reason I do them. Okay. Last time I got talking about the control of the Lac Operon, and when I finished, I was talking about how the Lac Repressor functions. So the Lac Repressor, you may remember, is a protein, and it's a protein that is binding to what I call the operator's sequence. Okay? Binding to the operator's sequence. And if we look at that, if I can find it. Nope. Nope. Okay, so if we look at what happens, so here was the beta-galactosidase gene. The operon, as I said, starts to in the yellow and moves to the right. This is a gene called the beta-galactosidase. This is a gene called the permease. This is a gene that has another function that I'll describe very briefly. Its total function is not completely known. And this is the operator sequence where the O is. The P stands for what your book calls the promoter, although, as I said, the two sequences actually overlap. The operator is bound by the repressor when there is no lactose present. When lactose is present, I told you a little lie, and for our purposes of this class, we can consider that little lie if it helps you to keep it in your head. And that is that lactose can bind to the repressor, and when it binds to the repressor can no longer bind to the operator, and so the promoter is left open and transcription can occur. Okay? The little lie is the fact that it's technically not lactose that binds to it. It's a modified form of lactose called allolactose. And allolactose is converted from lactose by the product of this enzyme. Okay? It's not a major point. If you think about it as lactose binding, that's all right as far as I'm concerned. Because the net effect is that when lactose is present, then it binds, the repressor is inactivated and transcription can occur. Okay? So allolactose, if we take a look at it, looks like this guy. This is the molecule that's binding. It's related to lactose. It's a modified form of lactose, and it is the actual molecule that binds to the repressor. Now, I mentioned last time that biochemists were lazy people, and that one of the things they like to do is they like to be able to measure something very easily by something like a color change, and so that's why I described XGAL. XGAL turns blue when beta-galactosidase acts on it. It turns out there's another molecule that biochemists use to study the Lac Operon, and it's called Iptg. It's a long name, and we'll just call it Iptg. I'll show it to you in a second. And what Iptg does is it also can bind to the Lac repressor. Now, Iptg is not lactose. It's not allolactose. In fact, it looks like this. Okay? It looks, however, enough, a portion of it that binds to the repressor looks enough like lactose and allolactose that the repressor will, in fact, bind it and transcription will be initiated. So the repressor can't bind to DNA when it's bound to Iptg. So Iptg is an artificial chemical. It's not a natural chemical. It's made by man. And it's given to bacterial cells to do this specific thing, to induce the Lac Operon. So here's a compound that we can use to actually turn on that switch And the advantage of using Iptg is that its concentration is constant. Cells cannot break it down. Cells can break down lactose. Cells can break down allolactose. And so if I try to induce the operon with lactose or allolactose and I want to make sure I've got expression going on, that expression is going to change if I use lactose or allolactose because it's going to get broken down over time. Iptg does not get broken down over time. Okay? So it's a way of turning on that promoter and leaving on that promoter. Very, very useful thing. Jenny?
Well, since that doesn't look very much like lactose,
it doesn't.
who thought of putting that into a cell to see if it would turn on?
That's a very good question. I don't know the answer to it. When we, I want you to keep in mind that when we look at binding sites on proteins, generally the entire molecule doesn't serve as the binding compound. So obviously whoever studied it or who designed this, and I don't know the story of the design of it, knew something about what it took to, for that binding site to bind this. But this thing works really, really well. It binds the repressor and shuts down the repressor. I'm sorry, it stops the repressor from functioning and allows transcription to occur very, very strongly. Okay, well, it may be surprising that when we add Iptg that the repressor works very, very strongly because if you recall from the last lecture, I told you that the Tata box that's present in the Lac promoter isn't a very good Tata box. And what I've told you about bad Tata boxes is they don't favor transcription very well. But I've told you when I get rid of the repressor, transcription goes great. Doesn't jive. There's a missing component. And the missing component is that the Lac Operon has another protein that can favor transcription strongly. And it's almost always bound to the region near the promoter. Okay? That protein is called CAP. CAP stands for cyclic AMP protein. Okay? Cyclic AMP protein. Let's see. So here's what happens when we have no allolactose, we have the repressor bound, transcription is turned off. Okay? So the operon is off when there's no lactose present. When lactose is present, the Lac repressor cannot bind to the operator, and therefore transcription can occur. Now, the piece that's missing is the fact that there is another protein that comes along, called CAP. There we go. I knew it was there. All right. CAP, as its name suggests, binds cyclic AMP. Now, when CAP binds to cyclic AMP, CAP can bind to DNA, and it binds to that promoter region. It turns out that the cell almost always has a little bit of cyclic AMP. So most of the time, CAP will bind to the promoter region. Now what CAP does, as it suggests here, is it binds and it favors, it actually helps the RNA polymerase to bind. This is how transcription becomes so strong. It favors the binding of the RNA polymerase. It helps it to get in there, and if it were just left to the Tata box alone, the RNA polymerase would not come in very strongly. But this guy is like a guiding thing, like maybe if you're doing a plane, okay, you want somebody to point you to the runway. This guy is pointing this guy to the runway being the Tata box where transcription is going to start. Okay? Now, that's what CAP does. As I said, CAP is bound most of the time, and it's bound most of the time because the cell almost always has at least a little bit of cyclic AMP in it. Well, the question then comes is, well, we've got a protein that positively affects transcription, and we've got a protein that negatively affects transcription, that being the Lac repressor. If we have them both bound, which one wins? What do you suppose? The repressor wins. And the reason that the repressor wins is the repressor, I'm going to orient you here, the repressor binds right here. And the repressor's binding site partly overlaps with the site that RNA polymerase wants to bind to. So when the repressor is bound, even this guy up here who's saying the runway is ready to go doesn't realize the runway is not ready to go because it's partly blocked by that repressor. The plane can't take off if there's another plane in front of it. And the plane in front of it is the Lac repressor. However, when the cell has lactose present, the repressor binds allolactose, it goes away, and now there's a place for the RNA polymerase to bind and transcription to start. Make sense? That was easy. You guys are an easy group today. Questions? Okay, that's good. So that's how the Lac repressor works. That's the actual structural site at the binding. And now what I want to do is start considering some things relevant to eukaryotes. What you've seen, the Lac Operon is an operon, and of course operons only occur in prokaryotes. I want to talk a little bit about eukaryotes. Eukaryotes have a very, very different set of considerations for controlling them, and again, we're not even going to barely scratch the surface. But we do need to think about the environment in which transcription has to occur in a eukaryotic cell. Okay? So let's think about eukaryotic chromosomes. Okay? In eukaryotic cells, we don't have nearly the situation we have in prokaryotic cells. In prokaryotic cells, for the most part, all we need is a binding site for the RNA polymerase, and it can take off and do its thing. Okay? The Lac operon is a little unusual in having an accessory protein facilitating this happening. Okay? In eukaryotic cells, we never have the RNA polymerase bind by itself. There's always factors that help it to bind, and this is trying to schematically show you that the assistance can be quite complicated. You look at this. There's several proteins that all are arranging themselves so they can facilitate the binding of RNA polymerase. Very, very kind of complicated situation that we have. We're not going to talk about all those proteins, obviously. Now, one of the reasons that we have to think about these problems is the nature of the eukaryotic chromosomes. If you remember, the eukaryotic chromosomes are composed of DNA and proteins. Okay? And the reason that they're composed of that is that the length of the eukaryotic DNA is too long to fit into the cell, let alone to fit into the nucleus. So we have to have proteins that help to coil up that DNA so that it will fit into the confines of the nucleus. And the proteins that we see that do that have a name. They're called histones. Histones. Histones are positively charged proteins that arrange themselves in very cool ways, as we shall see. And actually, you're seeing it partly here. I'll explain it in a minute. Okay? That facilitate the winding up of the DNA. The fact that they're positively charged allows them to interact very nicely with the negatively charged DNA, and they form a fairly tight packing as a result of that. So if I take a eukaryotic chromosome and I look at it in the microscope, it's a knot. It's a mesh of protein, DNA, all these tangles of tangles of tangles of tangles of tangles, so that I can fit this 7 feet of DNA into this small space. Okay? If I'm very careful, and I take one of those chromosomes and I very gently start pulling it apart so I can look at it, this is the result of what I see. It's something called beads on a string. And beads on a string are that sort of partly unwound chromosome that had all that knot of stuff that I was describing to you. We have to be gentle in doing this. But if we are very careful, what we see is this. You see that little gap right there? That's DNA. This little circle right here is DNA wrapped around a core of histones. Then a gap, core, gap, core, gap, core, gap, core. You get the idea. Okay? That core that's there is something I want to say a little bit about first. Okay? So the core is a structure called a nucleosome. A nucleosome is comprised of 8 proteins, 4 copies of 2 each of histones known as H2A, H2B, H3, and H4. H2A, H2B, H3, and H4. There's 2 copies of each of those, and they form what's called an octet. And this octet arranges itself in a circle, somewhat like what you see here. And that circle is the inner core of that bigger thing that I showed you. So here is the DNA wrapped around there. So we might have one end of the DNA coming in here, wrapping around a couple of times, and then it goes on out. That's basically what happens in a nucleosome. Okay. So a nucleosome, if you want a definition, a nucleosome is a repeating structure in chromatin. And now I have to give you another definition. What's chromatin? Chromatin is comprised of DNA and histone proteins. Okay? So a nucleosome is a repeating structure. So when the repeating structure is what I was showing you. Core, what's called linker, or gap, whatever, core, linker, et cetera, et cetera. So this repeating structure is called a nucleosome, and that stretches for a couple of hundred base pairs. Okay, these are all structural things. I'm not going to talk about that. If we look at the structure of the histones, here's H2A, H2B, H3, and H4. We see that they're very, very similar in structure. Some slight differences, but they're small proteins. They're not very large. And what's interesting about this is if we compare the amino acid sequence of histone proteins from the lowest eukaryotes like yeast to the highest eukaryotes like human beings and dogs and cats, what we see is that the structure is almost absolutely the same for all of them. And we also see that the amino acid sequence is almost identical. Not absolutely, but almost identical. Now, as we've talked about before, whenever we see something that does not change from one species to another, that means that that structure has to have a very, very important function, and there's not a lot of tolerance for change. If you mutate so that you interrupt this structure, that cell is not going to be viable. So very, very little change says that this evolutionary conservation of these tells us that these structures are very, very important, and the cell does not tolerate much in the way of change with respect to them. Well, of course, if all we did was we took and we wrapped around what I showed you earlier, the core, linker structures that are there, we would compress the DNA some, but we wouldn't compress it nearly as much as we need to. We need to compress the sequence, the length by a millionfold or so. Well, one of the ways that we do that compression is, here's that nucleosome that you saw before, written as this circle with the DNA wrapping around it. But then the individual nucleosomes themselves are arranged in ways that are tightly packed. And so we can actually imagine a coil of coils, as it were. And that's what we see. That's what really makes up a chromosome. A eukaryotic chromosome, that is. This is a mess, okay? Imagine that the gene that I want to transcribe is way buried right in here. How do I have to do that? Well, that's part of the reason why cells use things like transcription factors, and we'll see a host of factors that come in to favor transcription other than just RNA polymerase. This is a complicated structure, and this complicated structure has to be opened up in order for particular genes to be transcribed or for DNA replication to occur, for that matter. Okay. Let's see. Let's talk about enhancer sequences again. So when I talked about eukaryotic RNA earlier, I said that we found not only promoters, which is what we see in bacterial cells, but we also see other sequence elements related to the promoter called enhancers. Okay? Enhancers, you may recall, were sequences of DNA known to be involved in transcription, but that are located in widely varying positions in the DNA relative to the gene. In some cases, they might be thousands of base pairs away, ahead of the gene. They might be thousands of base pairs in front of the gene. They might even be in the middle of the gene. And that was very different than the simple promoter that we saw in prokaryotic cells. So how is it that enhancers do what they do? Well, enhancers, of course, enhancer sequences or enhancer sequence elements are bound by enhancer proteins. And those proteins, as I mentioned in class, are tissue specific. So we have certain cells will have certain enhancer proteins that will bind to, obviously, very specific enhancer sequence elements. And other cells won't have those. So, for example, if I'm a liver cell, I'm going to have some enhancer elements that are going to be binding to sequences that turn on gluconeogenesis, because the liver does gluconeogenesis. But my skin cells, which don't do gluconeogenesis, won't have those enhancer proteins. Okay? Well, enhancers, as we've talked about before, can have multiple copies of a sequence. And we frequently see multiple copies of them. This helps to facilitate the binding. More opportunities for binding if we have multiple copies of things. And what we see is that enhancer proteins bind to these sequences, not surprisingly, and they facilitate an opening up of the DNA. Now I'm going to come back to that, but I want to plant that idea in your head to begin with. They're going to facilitate the opening up of the DNA. Okay. Now, I like this slide a lot because it shows us something about how we can use some of the tools I've already described to you and to show something very graphically. And the thing that you're seeing very graphically is something that's very, very, very tissue-specific transcription and translation. This is a fertilized egg of a chicken. Okay? And the fertilized egg of the chicken has DNA that has been altered. It's been altered such that the promoter that is specific, that a promoter that is specifically made in, transcribed in muscle cells has been linked to the beta-galactosidase gene. Okay? So you go out and you say, okay, here's a protein that I make in muscle cells. Maybe it's creatine kinase. I've got the promoter for creatine kinase. I link the promoter for creatine kinase to the coding region for beta-galactosidase. Okay? So that what that means is that when that promoter, which would be active in muscle cells, is active, transcription of the beta-galactosidase will begin. Everybody with me? So only muscle-like cells will start making beta-galactosidase in this chicken. Well, now think about how lazy biochemists are, and they put xgal in there to measure how much beta-galactosidase is being made. What do they get? Well, wherever beta-galactosidase is being made, you start seeing blue. Now, look at this very early stage of development of this chicken, and we can see that there are already regions that are starting to behave like muscle cells. Because wherever the blue is, that's where beta-galactosidase is being made, and where beta-galactosidase is being made, that's under the control of a muscle promoter. Very, very cool things. This is hopefully reinforcing what I'm telling you earlier about how gene expression is going to be tissue-specific. It's going to vary from one cell to another. And we didn't see this whole thing being blue. We saw only specific regions that were going to become muscles in this chick being transcribing beta-galactosidase. Make sense? Okay. Yeah?
So how does the blue get there? Is that just a major molecule?
So the blue comes from the fact that beta-galactosidase, when it acts on xgal, cleaves it as if it were lactose, but it leaves a blue molecule, it leaves a blue product as a result of that. Okay? So it's like the thing I talked about last term where I had the chymotrypsin that cleaves something and gave us a yellow color. xgal, when it's cleaved by beta-galactosidase, gives a blue color. So only cells that are making beta-galactosidase will be getting this blue color. Well, not only can I look at this and say where these muscles are developing, I can also look at this and say, well, look, this muscle cell is making a heck of a lot more beta-galactosidase than this one is. Okay? This might be a heart muscle, this might be a skeletal muscle, etc. We could later track that and trace and see where, in fact, these early embryonic tissues end up in the chicken and learn something about them. It's really cool. Okay? So it's a very, very useful tool. This is one of, there are thousands of examples of the use of beta-galactosidase and XGAL to follow things inside of cells. Okay. Control of gene expression. Control of gene expression. Eukaryotic cells have many things that control gene expression. I'm going to talk later about something called histone deacetylases. And histone deacetylases facilitate things with those enhancers that I talked about. So I'm going to come back to the enhancers in a bit. Another factor that affects transcription in eukaryotic cells is whether or not cytosine residues in the promoter have been methylated or not. So there are enzymes that will put methyl groups on cytosine to make this 5-methylcytosine. Again, structure is not important. Make 5-methylcytosine, and when this is present, or there's a, I shouldn't say one, but a bundle of these present in the promoter of a eukaryotic gene will turn off transcription of that gene. Okay? Now, how many people have heard of epigenetics? Okay, so epigenetics refer to changes that happen literally to the DNA of a cell that don't change the sequence. Okay? And they can be transmitted in some cases across generational barriers. This is one example of something that can act epigenetically. It doesn't affect the sequence, but it affects whether or not a gene is being made. Okay? So this is a real good example of an epigenetic change that happens, and it has nothing to do with changing or mutation or anything like that. These sorts of changes can, in fact, be transmitted across generations. Okay. Estradiol. Okay, I've talked a little bit about this. Estradiol, of course, is an estrogen, female hormone. And estrogens, as I've talked about before, are hormones that are involved not only in female sexual development and reproduction, but also in signaling. And signaling sometimes in tumors can be a factor. Here's a couple other molecules that act as hormones. All-trans-retinoic acid plays a very important role in the development of cells. And here's thyroxine, which is a thyroid hormone. Well, what I would like to do right now is focus on the transcriptional control for a gene that is controlled by estradiol. Okay? Just one example of many examples we could talk about for different types of transcriptional control. So I'm going to talk about the transcriptional control of a gene that is controlled by estradiol. Okay? Well, it turns out that when we look at the control, one of the factors involved in the control is a protein that, not surprisingly, binds to estradiol. Now, in the simple scheme, we might think to ourselves, and by the way, the simple scheme is wrong here, but I'll tell you what we might think to ourselves, is that estradiol makes its way into a cell, it binds to the protein that's a transcriptional factor, and when it binds to the protein goes to the promoter, and it starts the transcription of that gene. It's a simple view, and it turns out it's not quite right. It's a little surprising. Okay? What they discover is when you look at the structure of this estradiol binding protein, okay, it's called a hormone receptor, by the way. What we find when we look at the structure of this is that there's a region of the protein that binds to the estradiol and a separate region that binds to the DNA. Okay? Not surprising. Two different regions that do two different things. It turns out, however, though, that the binding of the hormone receptor to the DNA does not, underline not, require the binding of estradiol. this is a little surprising. Okay? Here's a schematic of what happens. Here's that protein. You see it's a dimer. Here's that hormone receptor protein. This is the protein that would bind estradiol, but as you can see in this figure, it is already bound to the promoter sequence. So how in the world does it affect transcription of this gene? Well, it turns out that what this guy is doing is it's doing what's called recruiting co-activators. In the state in which it is shown on the left, where it has not bound to the estradiol, it cannot bind other proteins. Binding of the estradiol doesn't change the binding to the DNA in any way. It changes the structure of the protein that binds to other proteins. So binding of estradiol causes the hormone receptor to change shape and open itself up to bind other proteins called co-activators. So if you may think back to that very first figure I showed you today, or one of the first figures I showed you today, where we had the transcription factor, and I said there were a complex of factors then that helped the RNA polymerase to come and do its thing. We can start to see how these factors are going to pile on, and they're only going to pile on when estradiol is present. Now, if this is a gene that is involved in stimulating the cell to proliferate, and this is a cancer cell, we've got a strategy for an anti-cancer therapy. The strategy is, well, if we can find something that binds but doesn't cause this to happen, or somehow stops these proteins from being able to bind, we can stop that cancer cell from proliferating. Okay? And it turns out that tamoxifen does that very nicely. So we hear about the anti-cancer compound tamoxifen. Tamoxifen doesn't at first glance look an awful lot like estradiol, but in fact it will bind to that hormone receptor. And when it binds to the hormone receptor, the region that would bind the protein is blocked. So the co-activators can't block when tamoxifen binds to the hormone receptor. By the way, I keep calling it hormone receptor. It's actually called steroid hormone receptor. I don't know why I can't spit that out. But the hormone receptor I'm talking about is called the steroid hormone receptor. Okay? So tamoxifen is very, very powerful anti-cancer compound for tumors that are estrogen sensitive. It doesn't work on other tumors. If they're not estrogen sensitive, tamoxifen isn't going to do a darn bit of good. But if the tumor is being stimulated by estrogen, tamoxifen is a wonder drug. Not absolute, doesn't kill everything, but it does a very good job of reducing that stimulation to divide. Okay. That's what I want to say there. Yes, question?
Does tamoxifen affect other cells that may not be cancerous but are still sensitive to estrogen?
Does tamoxifen affect other cells that would be receptive to estrogen? And in fact, yes, it will. So women who are taking tamoxifen may, for example, go through menopause. Okay, and so it can affect any cell that would, in fact, have that estrogen stimulus. Okay, so that's one example of how transcription of a particular set of genes is controlled. Now I want to step back and look at a more general type of control that we see for many, many genes. Okay? And this general type of control is now going to come back and talk about those enhancers that I talked about earlier. All right? So for general type of, or general considerations for transcription, we have to remember that one of the things I said that was important was we have to open up this structure. Opening up this chromatin structure is very, very important, and there's several steps in the process by which this occurs. Okay? There are a set of enzymes known as histone deacetylases. Okay? Histone deacetylases. Histone deacetylases, and acetylase is spelled A-C-E-T-Y-L-A-S-E. A histone acetylase catalyzes the reaction that you see right here. It takes and it puts an acetyl group onto lysines. Remember I said that the histones are very positively charged. They're very rich in lysines. So if I have a histone acetylase, what it's doing is it's adding an acetyl group to that lysine, and it's covering up a positive charge. Notice we have a positive charge here on the left, no charge on the right. Now, one of the things that was favoring chromatin structure was the fact that the positively charged histone was attracted to the negatively charged DNA. It made a nice tight lock. Positive attracts negatives, right? If I take and I start covering up a lot of those positives with a histone acetylase, what's going to happen? Well, the bond or the linking between the histone and the DNA is going to be loosened. It's not going to be such a nice, tight little complex. It's in fact going to relax. And that relaxation is very, very important for opening up this structure. I'm going to show you some other considerations as well. But one of the first things that has to happen is that a histone acetylase has to open up, has to put an acetyl group onto lysine of histones. Okay, you with me so far? All right, so we've sort of started to loosen up this big, complicated, very tightly wrapped structure with a histone acetylase. Before I go forward, I want to also point out that there are a class of enzymes that do the opposite reaction. They're called Histone Deacetylases, or HDAC, HDAC. A histone deacetylase takes the acetyl group off of histones. And I'll ask you, do you suppose that would favor transcription or favor repression of transcription? Repression, right? Because now you're going to favor that complex becoming tight again. I'll tell you a fascinating thing we now know about some histone deacetylases. Some of them are linked to cellular longevity. There's a class of histone deacetylases known as sirtuins, S-A-R-T-U-I-N-S. And sirtuins in many systems have been linked to lengthening the life of cells. Okay? So turning off gene expression, okay, in a general sense, may in fact favor cells living longer, favor organisms living longer. Interesting thing. Okay, I'm not going to say more about that here. All right. Now, here is a histone H4 that has been acetylated. It's got its lysine has an acetyl group on it. Okay. There are proteins that have a structure called a bromodomain, B-R-O-M-O-D-O-M-A-I-N, A-I-N, okay, that recognize acetylated histone residues. So proteins that have a structure called a bromodomain recognize and bind to acetylated histone residues. This would be a very good quality for a transcription factor to have. Okay? A very good quality for an enhancer to have. And enhancers have bromodomains. Now, let's look in a bigger picture what an enhancer is doing. Okay? Remember, the enhancer is binding to an enhancer sequence element in the DNA, and that sequence element may be located in a wide variety of places relative to the gene that it is linked to. Here's a transcription factor that has just found an acetylated lysine residue in a histone. It has, in fact, bound to this sequence. Okay? So the transcription factor has come in. Actually, let me back up. I don't want to say that. The transcription factor hasn't bound. The transcription factor is binding the DNA. It's not binding the. It doesn't have a bromodomain. It does not bind to a protein. It binds the DNA. So the transcription factor is recognizing a specific sequence. This could be something like an enhancer recognizing an enhancer sequence element, and no, enhancers don't have bromodomains. I'm getting ahead of myself. So start over. A transcription factor, like an enhancer element, binds to, recognizes and binds to a specific DNA sequence, period. the enhancer protein may recognize, okay, that sequence element wherever it happens to be. But once it is bound to that sequence element, several things happen. The transcription factor is a target for binding by something called a co-activator. So the co-activator that we saw in the case of the steroid hormone receptor, here's another co-activator. Okay? It's simply binding to where this transcription factor was. If you want to think about this as the steroid hormone receptor, that's perfectly fine as well. In any event, this co-activator binds to that enhancer that was there, and the co-activator acts as a histone acetylase. So the co-activator, one of its functions is to acetylate lysine residues on the histones adjacent to where this binding has occurred. Okay? These acetylated lysine residues become targets for proteins known as the remodeling engine. The remodeling engine recognizes bromodomains. There we finally have the bromodomains coming in. So they recognize the bromodomains, and what the remodeling engine does is it starts stripping away the histones wherever it has bound to here. The fact that these guys have already been loosened up makes this guy's job a lot easier. And when it's done, you have an opened up region where RNA polymerase can get in and start moving. So the remodeling engine facilitates the opening up of DNA so that that transcriptional complex can get started doing transcription. Okay? Multi-step, a little complicated. Questions about that? I've got one more thing to say about it, and then I'll shut up. Jenny? So the proteins where the bromodomains are the histones. Everywhere we see one of these cores. So that's what's happened here. You can see that these proteins have gotten lysine residues acetylated. And so now this guy comes in and says, oh, they're lysine acetylated residues. There must be a reason to open this thing up, and that's what it facilitates. So now we start to see how that very complicated chromosomal structure starts to bear at least a short region of itself for transcription to occur. Are there logistical hurdles? Yes. Okay. Is it complicated? Yes. But those things are all there. Now, one thing you might wonder is, well, it is very complicated, but how did the transcription factor get in here in the first place? Okay? This was a very complicated structure, Kevin, and this transcription factor, this enhancer protein, had to come in and bind to a specific sequence. How did it get there? There's one thing I haven't told you, and that's what I want to tell you now. I want to go back to the beads on the string. Okay? So if we look at the beads on the string, okay, remember what we have here is we have a core, a linker, a core, a linker. This core has 8 proteins, and the DNA wraps in a circle around it. This guy out here, which looks like bare DNA, turns out not to be completely bare DNA. It is bound by another histone called H1. So H1 binds in the linker region. It probably plays a role in helping the cell to decide which regions are open, or at least relatively open, that is not wound. All right? So H1 is helping to tell the cell, keep this in a circle, keep this open as you can. This open region is the place where the transcription factor has to bind. Okay? So H1 is helping to play a role in keeping the sequences open that are necessary for transcription factors to bind so this whole process can get started. I see some frowns, which usually is not a good sign, so I should stop and take questions on that. Yes?
But the DNA is not like this. The DNA is coiled.
It is, okay, but nonetheless, there are still linked regions between them that are open. It is. So that coiled coil is hard to envision, but in fact, even the coiled coils have open regions within them. No questions at all? Pop quiz? Okay. Well, let's see. It's Friday. Why don't we just call it a day?
There must be some regularity between the distances between those sites where the transcription comes in for the histones to be spaced so regularly.
There is. One of the hot areas of investigation in chromatin science is understanding that spacing phenomenon and how it occurs, because it's pretty critical.